Hi, Clementine.
Hello, how are you?
So you're still in the UK?
Yeah, for now I am. I haven't found a job yet, so we'll see, but still looking. I'm just about to actually submit the final version of my PhD, which we have to do after our defense.
Okay.
Like you have
Well,
to, yeah, basically you have to do the paperwork after your defense.
well, at least you're now in an easier time zone for you.
Yes, yeah, definitely. It's poor Anthony who's suffering with the morning slot, I think. Don't know if he's coming today.
Yeah, and Dunya just texted that she's ill, so yeah, she has usually the latest update on which is tested and which not. So, but they are fully working on the lab, testing a lot of compounds at the moment. So,
Okay.
and somebody went to the Teams link, so I will
Oh,
direct them to the Zoom link.
Hi everybody, I'm just on school run and other things, so I'm on audio and hopefully can listen in as long as I can.
Okay. Well, at least you don't have to chair or take the
take
minutes,
minutes.
so that.
Yeah, exactly. Yeah, I checked that.
Okay. Just see if somebody will show up in the here now.
Are we expecting Erica? Um, Anyone know?
I'm not sure. Yeah, she did say she couldn't come. I'm not sure. Hopefully.
Yeah.
Because apparently it's somebody outside of our organization.
Mm.
So
I think Louisa is presenting at the ACS meeting at the moment, so may not be able to join depending on time zones.
yeah, I'm just checking somebody is in the Teams link, if I can see who it is. Oh, Max, are you also in the Teams link? Yeah. Okay, then I think we'll just start. Welcome everybody. Today it's the March meeting, and we hope to have a lot of nice updates on general business. We have now the LinkedIn page up and running. And I just posted last week that Mickey had his PhD defense. He defended his thesis with honors, so that's great. I posted that also on our LinkedIn with a link to this meeting. So I hope that maybe some people picked it up and would join. But yeah, I don't think there are many other people, new people here. So, but maybe if we do this more often, then at some point some new people will join. And I think that Max and Dunya worked on some short content. And Dunya is planning to do a day in the life of a mycetos scientist testing new compounds to make a short movie and
She
to post as well.
sorry,
Yeah,
she
Clementine?
sent me that video. I'm just trying to she's made the video and I've got it. I'm just trying to figure out how to share it, or maybe I'll just upload it if that's easier. But yeah, we've got that ready to go.
Okay. Yeah.
I had a
So
quick question about the thing that I think Max mentions on the issue, which is about invitations for people that we can send out. I'm not quite sure how that works. Anyone how that works? I'm not sure who owns those and how we should use them.
Yeah, there is something if you log into the.
Max cannot. We cannot hear you.
Oh, Max, we cannot hear you when you talk. But when I logged into the
now?
link, yes, Max, the floor is yours.
Great. Yeah, I made an issue on the GitHub where just as a place where we could maybe brainstorm some ideas for new posts, and I there also raised the point that we have 100 invites currently, and we could just try it out for someone within our own group. Maybe even have them unfollow the page if they already follow, and just send them the invite and look how pushy it actually is if it's a kind of a nice way to do it or if it's a bit obnoxious. But we have 100 invites to start, and then every month for the free account it should reset to 50 invites. So that's quite a lot of invites you can send people, and I think it will just be the MycetOS page, which will be a bit like a follower request, but just not they follow you now, do you want to follow them back, but more, hey, you want to follow me.
So what we could do, for instance, I could simply ask send an invite to my husband. So then we don't have to unfollow it ourselves. And then I can know from him how pushy it actually is.
Yeah, that might be smart.
Yeah, I mean that would be great. And if so is it the case who's allowed to send those? How does it work?
I'm not sure. At least I am able to send those, but I don't have a large network of people who would actually be very related to like a fungal diseases field. So
I can
I'm
also
probably
do
not.
this because
You
when
can
I
also do
put
it.
the content of Mickey on, I also got that notification that I'm able to send out 100 invites. So
probably it has to do something with the admin rights. So either because both Wendy and I are like administrators that can also manage the entire page, and the idea is that we should make at least everyone who wants that have posting and commenting rights for the page. So we should maybe figure out if you're also able to do that with the commenting and posting rights, and then anyone could just start doing that.
Yeah, because maybe it's also good if Matt gets admin rights then too, and
I
maybe
think he might
also
have admin rights already.
Yeah, and maybe somebody from DNDi, because they have a great comms team who can help us with that.
Yeah, sounds right.
Okay. Are there any other things we need to discuss about the LinkedIn page?
I had a little thought because there were two posts I think that we that did really well, at least reached a really broad audience, and were viewed by a few thousand people, which I think is pretty good for a page that has 50 followers. And those were the posts that were like reposted by some of our more high profile members. So I think that is something to be aware of, that is at least at the moment, now that the page is still a bit smaller, the way to get more people to watch the post. And also we
which
have
posts were those?
mostly the PhD celebratory posts.
Okay.
I'll have to check which ones precisely, but we mostly have PhD like announcements and celebratory posts, and one post of the World NTD Day, which like launched our page and highlighted one of our papers.
So maybe if when Clementine puts Dunya's content on, that if Matt, I, and some others who have a lot of followers repost this on their own page, that we attract a lot of people to the page.
Yeah, and we already have quite a lot of followers who are not part of the consortium. Like most of our followers are currently like from all over the world, from countries that are, I think, not in our consortium, and from a lot of endemic countries.
Okay, that's good. Today I'm speaking at the Riot Science Club here in Rotterdam. That's an open science club from the university where all kinds of open science projects will be promoted. I will also put a link for our own MycetOS page, of course, and also how to join. So hopefully maybe some people will join from there as well. And we will put that, I think I will put that same thing on the LinkedIn page, so then they can take it up from there. Okay, if that was all on the GitHub page, then maybe good to go to the active series. When I constructed the page or the agenda, I just thought it's good to go through the different series. So for the series 2, are there any updates since last month, Clementine?
Not really, but I have shared the experimental with, like, I've put it on the same issue where we've I guess been talking about characterization already. The only thing I would have to say, it's not really an update, but we were talking about the compounds that we would include experimental for, and I have almost everything for the compounds which are specifically mentioned in the paper. So, where we say like these ones were active and these more active than these and whatever else. I think I'm just missing a couple of those newest ones made by the Uni of York, the MyOS 901 to 906, maybe one other one. But when it comes to the compounds that aren't specifically mentioned or described in the paper, I don't really have any data at all for them. So I have some data for the ones that were made by UCED, but I don't really have any data for any that were made by UCL or York. I don't think there are that many made by York, actually. So I haven't been able to do anything with that part, basically.
Okay, Matt, do you think you can still have access to the data? Because I assume that they were characterized in your lab if they were made in UCL.
Yeah, actually, yes, it's the MSc students that make them. I will need to double check what we have. I'm sure we definitely have NMR, carbon and hydrogen, probably not IR and LCMS.
Well, I guess that's something to check, obviously, before we get around to submitting it.
No, I mean the ones that you are not including in the paper. Do you need the LCMS and the IR as well?
That's a fair question. I don't know. I think it depends on how easy it is for you to get the characterization together. If you've got the proton and carbon for all of them, it seems reasonable to me that we say these ones that we haven't actually explicitly discussed, we did make, here's the the basics kind of the characterization, but the other ones we've done more fully.
We've done more fully. Okay, so I'll gather the data and upload them on GitHub of everything else.
Yeah, that would be great.
Okay.
Okay. Other things we need to discuss about series 2?
And I think just that, unfortunately, those last compounds from York weren't hits. I can't remember if we mentioned that last meeting or not. Not quite sure. I think Junior was just finishing the testing last month.
Yes. So, yeah. I think she put everything on the spreadsheet, what we do have. And I know that Bora, the technician, is working on testing the final things we need to test. So, okay.
Yeah.
Then
sorry, Junior.
Yeah, and I
said
that,
none of them were a hit.
Yeah, and I see that Peter said that he will send the outstanding data this week. So the data you still miss.
Oh, okay,
and then
perfect.
for series 5, we also have to wait for the biological data. I didn't double check if the full data set is available already, but I will talk to the technician later on and double check if the data is there. If so, then I will make a note on GitHub, then we can start working towards completion of that manuscript. And then series 7, that's the most active series, of course, at the moment. There were some compounds already came in. Also, DNDi did send some compounds. I just got a confirmation by DHL that they are on the way. So 19 compounds from DNDi are coming in. So they are expected to arrive tomorrow. So we will have some additional compounds to test for that series. And I think there's also some biosynthesis planned. Has anyone has an update on that?
So we've just finished our year 3 students have just finished making some. We spent a lot of time trying to develop methods to make these compounds. And the long story short is that anything with electron withdrawing groups, which seems to be the current hit compounds, are not really able to be made by undergraduates that I've found in terms of the chemicals that you need to make them. But anything electron donating or aliphatic works for the coupling on the amine. So yeah, that's where we got to. We didn't make loads, but we've got a relatively robust method for making some of them, just not the others, is where we got to.
Okay.
So does that mean that, sorry, does that mean that we don't really have a way at the moment to make compounds we're most interested in? Or are there still ones that we're keen on?
Yeah, I mean, I think there might be the odd one or two that we could make. But the ones that seem to have the most activity at the moment seem to have that electron withdrawing groups on that ring. But yeah, I don't know what the long-term plan would be. But I guess if there's a whole host of compounds being sent to be tested, we might have a little bit more of a scope of what type of compounds we want to make. So I know that we tried to make the methoxy versions of them, which didn't work. But we've got a method to make the unprotected versions now, but only on the electron-rich species. So, yeah, that's where we're kind of at. But whether someone else could solve it potentially, I don't know. But we did try quite hard. So, don't know.
I mean, my guess is that if you guys can't figure it out, that you said first years are not going to be making those compounds.
Yeah, I mean, I think you can almost certainly make them if you're willing to use like PCl3 or thionyl chloride or something. But I'm not particularly excited about doing that with a load of undergraduates. But yeah, I think that's where we're at. We
can definitely put together some kind of call out to put on LinkedIn, for instance, to ask people for compounds, like, you know, as in to see who might have some compounds that we could test. I'm not sure beyond that. Long-term plans sounds like possibly we'll need to work on a new series.
Yeah, that was also what I was thinking. If this becomes too difficult for the students and you still want to keep the students on board of MycetOS, there's so many targets we never tested, and there are so many hits still unexplored, of course, in the master list. Maybe somebody could browse the master list to look for molecules which are easier to adapt and which had activity against Madurella mycetomatis. So it's not a problem if we, because we always worked with multiple series in the same run, and it seems that after we close series 2 and 5, we only are working on series 7 with chemistry. We could always include another series where the students could play a more active role, because it would be a waste, of course, to not be able to work with the students anymore because we chose a too difficult molecule to optimize further. And the more targets we explore, the more options we later have for combination therapy. So yeah, that might also be an option. So that would be a little bit weird selection point, of course, to see which are the easiest molecules, but still interesting molecules to make chemistry-wise. But maybe we stumble on something which we otherwise would never have thought of. So I'm not a chemist, so I won't be able to do this. I can only see which compounds are biological active. So maybe one of the chemists would like to take this task.
Yeah, I can have a look through. If someone points me in the direction of which place to get the data, I can certainly have a look at. Is it on the big spreadsheet?
Yeah, on the big spreadsheet we have those smiles.
Yeah.
I can of course not read them very well. I know what they are. That's how far I am at the moment. But you probably can.
Yeah. Yeah, we can I'll have a look through and see if we can highlight anything. But yeah.
Okay, then maybe we can discuss that either next month or the month thereafter, depending on how much time you need for that.
Yeah.
Anything else we would like to discuss for now on Series 7? If not, then we have the proteins which are made. So Matt updated us last month that they did express CytA from Aspergillus fumigatus. Saskia and one of our students, Atik, were working on the Madurella variant. Saskia, do you have an update?
Yeah, so unfortunately nothing for CytA yet. For the MKK, we checked, and both PRA, we checked the soluble fraction. And for PRA, it seems we have at least a clone that might express the protein. It is a different height than expected, but that's possible with the potential dimer forms maybe. For SKH, we didn't see anything, but we only checked the soluble fraction. So it may be in the insoluble fraction. We'll check that. So yeah, that's sort of the current status. So perhaps for PRA, yes, the other one's still ongoing.
Yeah, and the students did put CytA in E. coli, but we still have to check if the plasmid was taken up by E. coli, and if so, if it also expresses CytA. We had a failure by earlier last week where we didn't get the plasmid in. So if the plasmid is not in, of course, E. coli cannot express it. So, but we hope that like the CytA from Aspergillus, the CytA from Madurella will go straight forward. Because if it works for Aspergillus, I don't see a reason why it shouldn't work for Madurella. Maybe Erica or Matt has an update on the status of the Aspergillus variants you are expressing.
Yeah, I asked Yelena for an update, but I asked far too late, so she hasn't had a chance to respond yet. But when she responds, I'll post something on the issue. Um, I mean, I think it's looking really good. I think they've just got a bunch of other priorities at the moment, but hopefully soon.
Okay. So hopefully they can be shipped quite soon after each other to be screened, because it would also be good to verify any compounds if they are active against both.
Yeah, absolutely.
Okay, so that's also ongoing. And then the final point of the agenda was the thesis of Mickey Konings. He defended his PhD, and I added it to the thesis page. And I know last month we had a discussion, where should we put the thesis? I still couldn't find the one from Hang and Clementine. So what would be an easy spot on the MycetOS to put those theses? Because now I put them under output, and the issue, there's an issue at the beginning called talks, meetings, publicity. But where did you put them on, Clementine?
So I've put the other ones in the general start here repository. I think we talked briefly about this the last meeting. We currently, just like for everything, not just theses, we have a lot of kind of overlap, I think, between our different repositories. And we talked about, instead of having a different repository for different things like, talks, meeting, publicity, monthly Zoom meetings, etc., we could potentially reduce the number of repositories that we have and instead use the wiki and pages within one repository to organize everything, which obviously we'll need to kind of, discuss it a bit. I can probably put together a plan, and we can talk about it next meeting and see if people are on board with it. But my personal experience and my suspicion for other people is that when you look at open source mycetoma and you've got something like 12 repositories, obviously some people instantly see, oh, what other molecules to screen, and go to that. And then some people just don't realize that there are more repositories if they scroll down, and they don't see that one at all. And then we end up with people, posting about the same thing in different places.
Yeah.
So, yeah, I think it would just be simpler to have a repository that's like, this is where you go to find where you need to go, kind of thing.
Yeah. For now, I will put them with the others as well, so then it will be double in the GitHub page. But
yeah, I think that's
so
the best solution for now.
Everything is there when you look for it. But I couldn't find it myself, so I was trying to look, but
yeah, and I think
where I always paste it.
I had a remark kind of related to this. When I first looked at the GitHub page, I was a bit confused by the monthly Zoom meetings tab. That the roster is available as an issue, and I was not familiar with GitHub, and I was not able to find the issue. So this might just be a me problem, but maybe it should be a little more dummy-proof, explaining you're now in the code tab and that there's a tab at the top where you can press issues or something. Like it took me a while to figure out, actually. So might just be me, but might be worth adding a sentence there.
No, I think it's very confusing when you start.
Yeah, I see that somebody pinned the roster now on top, so it's also on top.
I think previously we've attempted to try and make it clearer, but it's always a bit difficult to figure out what's clear once you're used to it. So it'll be good to get your feedback when you're one of the more recent people to have gone through
Yeah.
trying to figure out how the hell GitHub works.
Yeah, I can just try to add a sentence to it to explain it, but probably if you're on the app or in the browser or whatever, the layout is all slightly different as well. So careful not to make it more confusing by adding very detailed steps that don't hold for a certain platform.
That's a good point, actually. I don't use the app. Well, I don't use the app for the wikis myself, but I can at least put a sentence somewhere that says, if this is not working properly in the app, maybe try the desktop version.
Yeah, I also never
I mean,
worked on the app, so yeah.
I believe
I have the app installed, so we can just double check if it still
Yeah.
works.
It definitely, because I used to use it because I write my like experimental notes on GitHub, I would use it all the time only for issues, not for any of the other stuff like pages and wiki. But I'm pretty sure they don't work that nicely in the app. Because yeah, every so often I would want to look up something on the wiki and it doesn't show up on the main page of the app, which is a problem.
Yeah.
Something to keep in mind.
Okay. So with that issue, it took me to the end of the agenda. So are there other things we would like to discuss for this month? If not, then we will, because by next month we will probably have some more biological data, then I will ask Dunja if she can make an overview of the latest biological data so we can take a look at it on a slide. Maybe that's easier than just going through this, okay, we're testing, then you can see how far we are with the testing.
That sounds good. And hopefully we'll have a bit of a progress on the papers as well.
Yeah, I hope so. Okay, then it was nice seeing you all again. For those who celebrate Easter, happy Easter for upcoming month. And then we will see each other in April again on the later time point for everyone.
All right, sounds good.
Great. Thanks very much, everyone.
Thank you.
Thank you.
Bye.
Bye.